Imagine a human brain, right? Perfectly sharp, fully aware, like functioning at 100% capacity, but it's entirely trapped inside a body where the biological wiring to the muscles just, well, it simply dissolves.
Yeah, it's an absolutely terrifying reality.
It really is. And
Yeah.
that is the reality of spinal muscular atrophy. And the most terrifying part of it all, the blueprint for this devastation is completely invisible on any standard medical scan.
Right, because we rely so heavily on the visible in medicine.
And exactly, like you break your arm, an x-ray shows the jagged line, done. But you step into the world of human genetics and suddenly you are navigating like the absolute definition of diagnostic muddy waters. The instructions that build us are invisible to the naked eye, yet they dictate almost everything about how our bodies function.
Because when you're dealing with the genetic level, you're trying to read a blueprint that is constantly interacting with the environment. It's sometimes masking its own errors and occasionally, and it's wild, occasionally rewriting itself spontaneously.
Which is crazy to think about. And that is exactly our mission today. We are taking a custom-tailored deep dive into your provided source materials to really demystify the architecture of human genetics.
Yeah, we're going to cover a lot of ground today.
We really are. For those of you listening, we're exploring what happens when these instructions go wrong, scaling all the way from massive chromosomal errors down to, like, a single missing gene. And we'll look at the specific tools from standardized pedigrees to direct consumer lab testing that medical science uses to track these invisible ghosts.
The ghosts in our DNA, basically.
Right. Okay, let's unpack this. Our sources for this deep dive include clinical fact sheets on chromosomal abnormalities, genetic disease classifications, a really deep look into spinal muscular atrophy, and the mechanics of genetic tracking. But before we hunt for specific typos, I think we have to look at how science categorizes this vast library of human disease.
Broadly speaking, clinicians organize human diseases into three main functional categories. First, you have diseases that are essentially entirely genetically determined. Second, diseases that are almost entirely environmentally determined.
Right.
And third, there's this massive category of multifactorial or polygenic inheritance.
Multifactorial. Now, in the past, people love to use that old baking recipe cliche for this. Your genes are the flour and sugar, your environment is the oven.
Oh yeah, the classic baking analogy.
Right, but for an audience that actually follows genetic science, I think a much more accurate mechanical analogy is a loaded spring.
Oh, I like that.
Yeah, so your genetics provide the tension.
Yeah.
You might inherit a tightly coiled spring for, say, hypertension or diabetes mellitus.
Right. The potential energy for the disease is physically there in your cells.
Exactly. But your environment, your diet, your lifestyle, your stress levels, that is the trigger. If
Yeah.
you never pull the trigger, the spring never snaps.
The loaded spring is, well, it's a brilliant way to conceptualize multifactorial inheritance because it really emphasizes that the genetic component is a susceptibility, right? It's not a guarantee.
Right.
Now, if we filter out the environmental and multifactorial stuff and just focus strictly on the genetic disorders, the medical field divides those into classical and non-classical buckets.
Okay, classical and non-classical.
Yeah. Classical genetic diseases include the single-gene Mendelian disorders, the massive chromosomal or cytogenetic disorders, and those multifactorial diseases we just mentioned.
And non-classical, because I imagine that's where the inheritance rules get a little, I don't know, strange.
They do. They really do. Non-classical diseases are still single-gene disorders, but they exhibit highly atypical patterns of inheritance.
Like what?
Well, a prime example from the clinical notes would be diseases caused by mutations in mitochondrial DNA.
Oh, right, because that's not in the nucleus.
Exactly. It's DNA, but
Yeah.
it's not in the nucleus of the cell, and it absolutely doesn't play by standard Mendelian rules. If we connect this to the bigger picture, organizing a patient's symptoms into these specific buckets is really the only way a doctor knows which investigative tool to deploy.
Because you wouldn't use a magnifying glass when you need a wide-angle lens.
Exactly. It's all about scale.
Well, let's pick up that wide-angle lens first. Classical chromosomal disorders. We're talking about the macroarchitecture of our DNA. What physically happens when these massive structures have errors?
So, looking through that wide-angle lens, chromosomal abnormalities generally split into two distinct mechanical failures. You have numerical and you have structural.
And numerical is just, it's simply a counting error, right?
Pretty much, yeah. We're supposed to have our chromosomes in neat pairs. If an individual is missing one chromosome from a pair, the condition is called monosomy.
Okay.
If they have more than two, which is usually three, it is a trisomy.
Down syndrome is the textbook example of a trisomy, right? Trisomy 21.
Yes, three copies of chromosome 21 instead of the usual two, and having that massive amount of extra genetic material disrupts typical development. It's marked by characteristic facial appearance, poor muscle tone or hypotonia in infancy, learning difficulties, and mental disability.
And what about the other side? The monosomy.
Right. On the flip side, a primary example of monosomy is Turner syndrome. That's where a female is born with only one X chromosome.
Just one. Wow.
Yeah. The biological impact there typically involves shorter stature and an inability to have children.
So numerical is a pretty straightforward counting error. Too many or too few. But the structural abnormalities are where the chromosome count is perfectly fine, 23 pairs. But the physical architecture of the chromosomes themselves is basically wrecked.
Yeah, that's where things get really complicated.
The sources list deletions, duplications, translocations, inversions, and rings. I want to focus on translocations because honestly, it sounds like genetic material is just teleporting to the wrong neighborhood.
Teleporting is a really vivid way to picture it, but physically it's a structural transfer. So a portion of one chromosome breaks off and attaches to a completely different chromosome.
Just completely jumps ship.
Yeah. There is the reciprocal translocation, where segments from two different chromosomes just simply swap places. They trade parts. But the more dramatic mechanical failure is the Robertsonian translocation.
How does a Robertsonian transfer differ from a simple swap?
So in a Robertsonian translocation, an entire chromosome physically attaches to another one at the centromere.
The centromere, that's the center point.
Yeah, the centromere is that pinched central waist of the chromosome. So you essentially have two distinct chromosomes fusing together at their anchor points to form one massive, structurally altered superchromosome.
It's literally like two cars getting welded together at the bumpers while driving down the highway.
That is a terrifying image, but yes, very accurate.
And then you have inversions where a piece breaks off, flips completely upside down, and reattaches.
Right.
And rings, where a broken segment of a chromosome folds back on itself to form a closed circle. And it sometimes loses critical genetic material in the process. Now, I want to challenge an assumption here.
Go for it.
If a child is diagnosed with a Robertsonian translocation or a ring chromosome, I think the immediate assumption most people make is that the parents passed down a broken gene. But that's not always the case, is it?
No, it's not. In fact, it is one of the most pervasive misconceptions in clinical genetics.
Really?
Yeah. Most of these massive chromosomal abnormalities actually occur as random, spontaneous accidents in the formation of the egg or the sperm. The parent's cellular architecture is perfectly typical.
Oh, wow. So they're completely fine.
Completely fine. We call these de novo mutations, meaning they're entirely new to that specific individual. The mutation is present in every cell of the child's body, but it was not inherited from a parent's broader genetic makeup.
It's just a random glitch during the initial creation of that one specific cell.
Exactly. And sometimes the glitch happens slightly later. If the abnormality occurs a few cell divisions after conception, it leads to mosaicism.
Mosaicism, like a mosaic piece of art.
Right, because it means only a certain percentage of the cells in the resulting individual's body will carry the abnormality, while the rest are standard.
That is fascinating.
It really is.
Yeah.
However, because structural errors like translocations can occasionally be carried silently and inherited, standard clinical practice dictates performing chromosome studies on the parents of an affected child, just to be absolutely certain they aren't carrying a balanced translocation themselves.
Better safe than sorry. So we've seen the wide-angle scale. Entire missing chromosomes or massive fusions at the centromere. Now let's put down the wide-angle lens and pick up the magnifying glass.
We're zooming in.
Yes, we are transitioning from missing whole chapters of the biological blueprint to hunting for a single microscopic typo, a classical single gene disorder. And this brings us back to the disease I mentioned at the very beginning of the show, spinal muscular atrophy, or SMA.
Yeah. Cytogenetically, this typo is hyperlocalized. It sits in the 5q13 region of the chromosome.
Okay, 5q13.
The structure of the chromosome itself is totally intact, but zooming in, we find a deletion of a specific gene, the survival of motor neuron 1, or SMN1 gene. In a patient affected by SMA, both copies of this gene are missing.
See, this is what blows my mind. The human body has tens of thousands of genes. Why does losing this one specific SMN1 gene cause the entire muscular system to progressively collapse?
It comes down to cellular biochemistry. The SMN1 gene's only job is to code for the SMN1 protein.
Right.
So when you delete the gene, that protein just vanishes. And the absence of this specific protein causes massive splicing defects in motor neurons.
And what does that look like physically?
Physically, it leads to the targeted degeneration and death of the anterior horn
Oh,
cells.
yeah, anterior horn cell.
Yeah, these are the lower motor neurons located within the spinal cord. They basically act as the final relay station before nerve signals exit to the body.
Yeah.
It also destroys the brainstem nuclei.
Here's where it gets really interesting. Think of the nervous system like a massive telephone network.
Okay.
You have the brain, which is the caller, and you have the skeletal muscles, which are the receivers. In a patient with SMA, the caller is fully aware, they're frantically trying to dial out, and the muscle is physically sitting there waiting to ring.
Yeah.
But that telephone wire connecting them, the anterior horn cell, is fraying and snapping because it lacks the SMN1 protein required to maintain its physical integrity.
The signals in the brain simply hit a dead end at the spinal cord. And human biology is ruthless with its energy efficiency. If a skeletal muscle isn't receiving signals and actively contracting, the body just stops supporting it. The muscle tissue wastes away. It atrophies.
Clinically, what is the physical progression of that atrophy? Like, what actually happens to the patient?
Because the motor neurons are dying systematically, the patient experiences symmetric and progressive muscle weakness. And this isn't just an inability to walk. The weakness affects the entire somatic system. You see severe poor weight gain and growth failure.
Oh, man.
Yeah. As the intercostal muscles of the chest wall weaken, the patient develops restrictive lung disease. The muscles supporting the skeletal frame fail, leading to progressive scoliosis, and the joints eventually freeze into permanent contractures.
But the defining cruelty of SMA, the thing that makes it so psychologically devastating, is the cognitive profile.
Yeah, the clinical texts emphasize this point heavily. Patients with SMA have completely normal cognition.
Normal cognition.
The disease specifically targets the motor neurons, leaving the cortical networks completely untouched. The brain remains entirely sharp, intelligent, and trapped inside a physical shell that is actively dismantling its own wiring.
Which brings us to the inheritance problem. If SMA requires a patient to be missing both copies of the SMN1 gene, that means two healthy parents somehow handed down a ghost gene.
Right.
Now, if you are listening to this and you follow genetics, you probably already understand the basic Mendelian math here, the 25% chance of an affected child when two carriers meet. But the real mystery to me is how medical professionals tracked these silent carrier genes across generations before the invention of modern genetic sequencing.
They track the invisible by mapping the visible outcomes. They utilize pedigree analysis.
The classic family tree.
Exactly, but a pedigree is a highly standardized clinical family tree.
Right.
What's fascinating here is the sheer power of this standardization. A genetic counselor in New York, a clinician in Tokyo, and a researcher in London can all look at the exact same geometric map and instantly read the flow of a recessive mutation across a century of a family's history.
And the symbols are brutally pragmatic. You've got squares denoting males, circles denoting females. A diagonal line slashed through a symbol means the individual is deceased. Vertical lines map the biological descent from parent to offspring. But, and I have to point out a hilarious clinical distinction from the source text regarding the horizontal lines. A horizontal line connecting a square and a circle indicates a mating or a relationship.
Yes.
The medical notes explicitly clarify that biology does not care about a formal marriage certificate. That horizontal line simply indicates a DNA exchange.
Biology is fiercely indifferent to social constructs. It just cares about the genes. But structurally, the most critical symbol on any clinical pedigree is often a single arrow pointing to one specific square or circle. That arrow designates the proband.
The proband, that's like the catalyst, right?
The catalyst, exactly. The proband is the specific individual who first brings the family's genetic history to medical attention. It is usually the first child to show symptoms of a disease like SMA. Placing that single proband arrow on the map allows clinicians to work backward. They trace the horizontal and vertical lines to suddenly illuminate a hidden network of silent carrier status that has been moving completely undetected through the family for generations.
It's like turning on a black light in a dark room.
Yeah, exactly.
But at the end of the day, even the most meticulously drawn pedigree is still just an educated historical guess. To actually verify the missing 5q13 region, or to confirm Robertsonian translocation like we talked about earlier, science requires physical microscopic evidence. And that brings us to the modern era of genetic testing.
Right, because genetic testing is the physical laboratory analysis of DNA, RNA, chromosomes, proteins, and protein metabolites to concretely identify these heritable variations. We've moved so far beyond just the pedigree. Today, clinical genetic testing is available for over 2,000 different diseases.
Over 2,000. And accessing that genetic material doesn't require a massive surgical intervention. It's shockingly accessible. We're talking buccal swabs, literally just rubbing a cotton swab against the inside of the cheek, or spitting into a tube for a saliva sample, or a standard blood draw.
That's incredibly non-invasive now.
Yeah. The sources mention testing for single gene disorders like Huntington's disease or Fragile X, but there's a specific methodology highlighted called GWS, Genome-Wide Association Studies. How does a GWAS actually work mechanically compared to, like, a regular single gene test?
So a single gene test is like looking up a specific address in a directory to see if the house is missing.
Okay, very targeted.
Right. But GOS is entirely different. Instead of looking for a known typo, GOS scans the entire genomic landscape across thousands of individuals. It looks for tiny genetic markers, basically variations in the code that show up more frequently in people with a specific disease than in people without it. It helps scientists locate susceptibility genes.
So it's pattern recognition on a massive scale.
Exactly, and finding those susceptibility genes is revolutionary because it leads researchers to entirely unsuspected biological pathways.
Wow.
Yeah, it shows us how a disease might be operating under the surface, which is really the first crucial step toward understanding and eventually treating complex multifactorial disorders.
But the source material also notes a massive cultural shift here. Genetic testing isn't locked behind the sterile doors of clinical research anymore. It has gone DTC, direct to consumer.
Yeah, people are doing this recreationally.
Right. They are ordering kits online, swabbing their cheeks in their living rooms, and getting emails a month later detailing their entire genomic profile. So, for you listening, what does this all mean? Like, if someone's direct consumer test highlights a susceptibility variant for a major disease, does that mean their fate is sealed?
Not at all. And this circles perfectly back to our very first topic, the classifications of disease.
Oh, right.
Having a genetic variant that is statistically linked to a disease does not mean it is the sole deterministic cause. You have to remember the loaded spring of multifactorial inheritance.
Right. The tension is there, but the trigger hasn't been pulled.
Exactly. Your direct-to-consumer test might tell you that your biological spring is tightly coiled. But if your environmental factors never pull the trigger, the disease never expresses itself. The data is a tool for awareness, rarely a crystal ball.
The speed at which this field is moving is just staggering. In this deep dive today, we started by looking through the wide-angle lens at massive structural chromosomal collisions. We zoomed all the way down to a microscopic deletion in the SMN1 gene that causes the heartbreaking physical isolation of spinal muscular atrophy.
Yeah, it's a huge shift in scale.
It is. We explored how clinicians map these hidden ghosts using standardized pedigrees and the Proband arrow, and finally, how a simple cheek swab and a GWAS scan can decode the deepest blueprints of who we are.
We've moved from guessing at shadows in a family tree to reading the raw code of human construction. This raises an important question, though, especially regarding that shift to direct-to-consumer testing.
What's the question?
We are rapidly entering an era where millions of people casually hold the raw data of their own unexpressed genetic typos, their silent carrier statuses. If society reaches a point where everyone suddenly knows they are a silent carrier for a recessive devastation like SMA, how will that alter the fundamental way we choose our partners? Or, taking it even further, how will it change the way we view our own human imperfections once we have laboratory proof that biologically none of us are truly flawless?
A profound and honestly slightly unsettling thought to leave you with. Thank you for joining us on this deep dive into the architecture view. Keep exploring those blueprints, and we will catch you next time.